So welcome everybody again to another episode of the ideaXme show, the show that profiles the humans behind the really big ideas that are shaping our world and inspiring future creators. And for all those that like really great stories, I'm Ira Pastor, your health, aging, and longevity ambassador along for this ride. So for the last few shows, we've been spending a lot of time journeying along the biologic architecture of life and aging, spending most of our time somewhere between the organism as a whole, its physiological networks, processes, and the world of the cell and its various macromolecules, the proteins, the nucleic acids, the respective metabolic architecture of the genes, gene regulatory networks. Borrowing a line from Craig Venter, we humans are not giant amoebas. We're substantially more complex. The human body contains on average something in the range of 30 to 50 trillion cells. Each one of those cells contains 25,000 protein-coding genes, and the cells execute literally hundreds of thousands of biochemical reactions per day. And for the most part of our life, from the point that we're first conceived through the processes of ontogenesis, development, growth, metabolism, repair, regeneration, and so forth, it works close to perfect. The big unanswered question, of course, is how. How this 50 trillion cell organism does things so perfectly through multiple decades of our lives. Most thought leaders agree it's not just random. So today's guest, who I'm extremely excited about, who is going to take us further along this theme and into some research areas we don't normally hear too much about in the sort of the traditional biopharma space, is Professor Irena Cosic. Professor Cosic, who is Emeritus Professor at the College of Science, Engineering, and Health at RMIT University in Melbourne, Australia, is creator of the Resonant recognition model of molecular interactions, which proposes proteins and other macromolecules and their interactions are based on highly specific resonant electromagnetic energy transfer. And this has opened up a whole new realm for really understanding biologic processes and potentially interfering and modulating them. With a bachelor's, master's, and PhD in biomedical engineering, University of Belgrade, Professor Cosic has served as the Deputy Pro-Vice Chancellor of Science, Engineering, and Health for the last few decades. She has lectured and consulted extensively and published extensively on subjects as diverse as molecular bioelectronics, bio-electromagnetism, molecular modeling, protein engineering, biophysics. The list really goes on and on. She runs a consulting firm for biotech companies called AMALNA Consulting, and she's truly published extensively over the years and serves also as a referee for a number of international journals, including the International Journal of Theoretical Biology, Protein Engineering, and APSM, and is involved in the initiation of actually a new journal with the Institute of Electrical and Electronic Engineers, focused on tissue, cellular, and molecular engineering. With all that, Dr. Cosic, thank you so much for taking the opportunity to join us on the show today.
Thank you, Ira. That was very nice introduction, and I don't know where to start from.
Well, why don't we start off? We typically start off the show with our guest, really just giving you the floor so people can learn about you. Basically, your background, where you grew up, how you first got interested in science, how you got into bioengineering, and ultimately your journey to how in 2019 you are at the forefront of an extremely, I'll say, unique and cutting-edge field in the biosciences.
Thank you. Then I will start from the beginning. Since I was a little girl, I was always interested in nature, how it works, what are those forces moving nature around. But because I grew up in engineering family, I was also very much acquainted with engineering from very early childhood. So when it came to going to the university, I decided to combine those two interests and studied electrical electronic engineering as well as physics. Through engineering and telecommunications, I became fascinated with resonances. You know that for example, when you turn on your radio, you don't hear all stations that are in the air. Mm-hmm. You hear only one that your frequency is tuned on, and that's your resonance. So it's very selective. Mm-hmm. And of course, I've done some work in telecommunications, but I found that biology and the human body at most and biological beings are so much more interesting, mm-hmm. because telecommunication is made by humans. So it's all predictable. We know how it works, but we actually don't know how biology works. And I was pretty sure that resonances do have some role in that. I started my biomedical engineering studies and even a career through analysis biological signals like brain and heart and resonances in those signals. But once when I had a chance to speak with people who are working with proteins and DNA and realized that the big problem is to understand how proteins and DNA, those long molecules, build up of 20 different amino acids, for example, for proteins, mm-hmm. and for nucleotides for DNA, have such a precise and specific activity. Biologists, biochemists analyze it as a sequence of letters and try to find out the similar patterns in those letters. But obviously that was not how it works. Mm-hmm. So being somebody from telecommunications, being somebody who is interested in resonances, I saw it as a signal and started analyzing this signal using standard telecommunications methods, okay. like Fourier transform. Of course, I didn't use just the signal as sequence of letters. I used the distribution of free energy electrons across the protein or DNA, which is a actual physical value. And I was surprised even when I did my PhD, which was on that topic, that this distribution of free electrons, energies along the protein and DNA have a specific frequencies for specific biological function. It started about 35 years ago, but of course no one believed. So it took about a few decades to prove the idea. And what even more, I found that this specific frequency for example, protein function, mm-hmm. is the same as the target for this protein. So it's like they interact on the same frequency, which is actually resonance. And then I introduced charge transfer to the protein. So I calculated that these frequencies are actually electromagnetic radiation that can be produced by electrons traveling along protein or DNA and producing electromagnetic frequencies which are same as the frequency of distribution along the protein. In last 33 decades or so, I've done many examples. They have been tested experimentally, mm-hmm. both in biochemistry and also in physics, where we managed to activate proteins only by electromagnetic frequencies. Mm-hmm. In biochemistry, we were able to design peptides or proteins with this specific frequency, which will have desired biological function. So it has opened a completely new area in biochemistry, biophysics, and practically how biology works.
It's truly fascinating when you think about just the complexity of the system that we have thought of today, and how we've sort of hung out at this level of sort of the wet and warm area of biology, but now realizing or well, you've realized it, for many decades now, and obviously you published your seminal paper back in the early 1990s on resonant recognition model biomolecular interactions.
Yeah.
But here you have literally orders of magnitude of other, I call small stuff or smaller areas of physics and so forth that we just have not explored. And there are, we can't just sit around and think it's just it's all random stuff. There's models and patterns. What are, we can go into, because you sent me some wonderful papers that you're working on now along the lines of aging and telomeres and things like that, which are very important subset to the show. But can you talk a little bit, and no, obviously no confidential stuff, but you have, aside from your work at the university, you also consult for pharmaceutical companies. What are some of the resonant recognition model targets that people are interested in? Is it broad? Are you looking at cancer? Are you looking at cardiovascular disease, neural disease? What are some of the things that you see, what your interests in research specifically, and what you see some of your partner companies interested in?
Thank you, Deb. Actually, you're right. The main model was published in 1994, and it hasn't changed much. But it was applied now to many different areas. Mm-hmm. And also we did a lot of biophysical proofs that the model worked. Mm-hmm. And that's where all these publications came. The main interest, of course, is cancer, mm-hmm. because that's such a puzzle. Cancer cells are the same as our cells, but they just don't differentiate. They don't grow properly. They just grow without any rules. So where did they lose the rule? I've done a lot of work in cancer, and interestingly, I found frequencies that are related to formation of cancer, mm-hmm. and also frequencies that can counteract it. Mm-hmm. So, for example, you know about oncogenes and proto-oncogenes.
Sure.
These are proteins. Proto-oncogenes are normal proteins, and they don't cause cancer. They're used in cell growth, normal cell growth. Oncogenes are slightly mutated proto-oncogenes that actually produce cancer. There are very small difference between those proteins, but they have two different frequencies. So we actually at RMIT University, we did design some peptides that can counteract oncogene activity, mm-hmm. and we got experimental results in the biochemistry in a petri dish that those peptides can destroy cancer. Mm. Of course, there is further funding that needs to be done and tag them. One of my very early things that I've done was on HIV when it was a big problem at that time. And I designed, firstly, I found that although HIV virus has such a big variability, so it's very difficult to produce vaccine, it has always the same frequency. So it means it always does the same function. Doesn't matter that sequence is different. So I designed peptides that can actually produce this frequency without producing HIV, but can induce immune reaction to this frequency and actually to the virus. This has been tested experimentally in rabbits in France, in CNRS, and there is a patent on this work. Mm. But more recently, as you said, aging is another interest of mine. And the papers that I've sent you recently are about analysis of telomeres, mm-hmm. and telomerase and progerin.
All right.
Sorry?
Right.
Yeah. Telomeres are the endings of our DNA. As we age, they are shortening. Telomerase is a protein that can elongate that. So they work in an opposite waves. And progerin is a protein that actually kills telomeres, and even young kids who have that protein, they can age in 10 years completely. Interestingly, those three macromolecules have the same frequency.
Wow.
And that's where we come to the opposite interaction, because two proteins or two macromolecules to interact, it seems they have to have the same frequency but opposite phase. So you can play with the phase as well when you would like to have an opposite reaction. So can you imagine, we have a frequency of aging. And if you can produce anything against this frequency, we can, who knows, maybe elongate our lives or keep us young for a long time.
That's a very obviously important topic, a very interesting topic, especially with everything that's going on in the longevity biotechnology community nowadays. But once again, they're purely dealing with the sort of the cellular level of things, but to bring in the electromagnetic dimension of it could offer a new really way to think about how the process works and ultimately how to interfere. You had me quite interested in something you just said sort of about and I'm not an expert in sort of electromagnetism, but you talk about sort of a phase and then a reverse phase to counter something and so forth. It's a very interesting, I come out of the sort of traditional pharmaceutical industry, but it is, over the last few years, it's been a very interesting time in seeing, some, like I point my former employer, I used to work several years ago for GlaxoSmithKline. And, they're a traditional drug company that makes traditional drugs. But back in 2015, I believe it was, they invested, I think it was about $750 million into so-called electroceutical research. So very similar, this concept that, we don't need this drug molecule per se involved if we can record, mimic, whatever you want, what the proper terminology is, this wave, and do the same thing. Can you give us some of your thoughts on electroceuticals, bioelectric or biomagnetic information, like transfer as a form of therapeutic possibility versus, the old school macromolecule, putting a macromolecule in? What are your thoughts on this evolving space, and where do you see it going?
I think that's the way to go. With my background in research I'm doing, I'm absolutely sure that's the way to go. Our bodies are working on electrical and electromagnetic energy. From brain, from heart, from muscles, it's all electrical, electromagnetic. But now with my research, we have shown that it's also electromagnetic at the level of proteins, at the level of DNA, at the level of cell membrane. So that's definitely way to go. And I just would like to give you an example that what we've done. We published that paper recently. I don't know how much you're aware of. There is a certain illness among Amish people, a lack of protein that can digest bilirubin.
Okay.
And there is no cure for that. So they can't live long lives. The only cure is to spend whole night under the blue light. We found that particular protein that they are lacking has a frequency within the blue light spectrum. So that's absolute explanation why blue light can replace lack of the protein and can cure them. I probably didn't mention the whole spectrum of our protein activities is from far infrared, visible, and a bit of ultraviolet. So it does encompass visible light. And that's the best example where we have shown the light, or actual electromagnetic radiation of specific frequency, can replace activity of the protein.
That's truly fascinating.
That's fascinating. That's really fascinating. We also manage in the laboratory to activate like I can just remember, to activate some proteins with specific frequency of light, but that was in the laboratory. But this particular example is how you can heal people with light of specific frequency.
Completely fascinating.
So you got me. I think that's the way to go. But the problem is still that we don't know frequencies to use. We don't know which electromagnetic radiation to use. And I'm sure that pharmaceutical industry does it in the same way as they do with drugs. They try, and then just occasionally find something that works. The thing we can actually calculate which frequency to use with which illness, so on. The only problem is also with skin, because not very many frequencies penetrate. Yeah, that's a big problem.
I think about that
and
one.
Cell membrane as well. So if you need something inside the cell, it's also the problem. And there are a few solutions that we work with biotech company that we work with to overcome this problem. We also did some work with stem cells.
Okay.
And that's very much related to work with cancer, because stem cells, you need to grow and differentiate them. So you don't want stem cells only to grow and replicate and to make a cancer, but you want them to grow and become the certain tissue that you want.
Sure.
And for that, you use now some proteins, but we can also use frequencies for that. We just got recently a paper accepted, which will be published. Maybe I'll send it to you in a few weeks or so. And that works on that area.
You know, you just hit another chord in my mind for a second there when you were talking about, cells and membranes. Does the resonance recognition model, as you work with it, also apply to, and I apologize if this is a strange, I don't know if I'm phrasing this correctly, but I'm thinking to the non-macromolecular things that go on in the organism. So I think of things like shear stresses and forces of mechanotransduction and all these other pieces of sort of the puzzle of life. Does electromagnetic, and maybe it's not just one frequency, maybe, when you're talking about a wound or complex regeneration as we see it, maybe it's many frequencies that do something. But does it apply at these different levels of what I'll call the biologic architecture of the organism, or is it purely at the gene protein level?
As I mentioned before, we work on electromagnetic basis, and you're right, from nerves to muscles. Resonant recognition model is specifically for macromolecules.
Okay.
But I have done a lot of work with nerves, brain, and even acupuncture.
Okay.
If you know, acupuncture is based on electrically conductive pathways through the body. These frequencies are also resonant, but they are in a completely different frequency range. Because as bigger thing is, the frequency is lower, because you have a lower wave. So nerves and muscles and brain are in a low frequency range, while molecules are in a very high frequency range. So that's how they separate their activity. But I have done a lot of cell membrane receptors, like, for example, receptors for pain. And we found how we can mediate pain if we change electrical or electromagnetic waves in the pain receptors.
Okay.
For another company, we did a project that's published, so I can talk about this. They use patches made of titanium salt patches.
Okay.
And that structure of the patch was made in such a way that it is resonant with the frequency of pain produced by cell membrane receptors for pain. So it is possible to interact with molecular activities from outside with something that will be resonant with molecular activities. There is a lot of other electromagnetic activities in the body that are of the other frequency range, and they don't belong to resonant recognition model, but I did work with that as well.
Thinking, so you had me in the direction of sort of larger stuff. Now I want to go even a little smaller, and I have a paper here of yours entitled "The Quantum Foundations of the Resonant Recognition Model," and clearly, completely over my head. But where I want to go with this one, so about a couple of months ago, we had John Joe McFadden from the UK on talking about quantum biology. As I try to sort of wrap my head around some of this stuff, when you get into sort of the realm of, well, how does all this quantum biology stuff potentially work? A lot of it, sort of the hypotheses papers focus back on areas of electromagnetism that are extremely esoteric to me. I've read papers on spin chemistry. I have read papers on something called the Zeeman effect and splitting electromagnetic waves. Really, things that only a handful of people on the earth, probably you, understand. Could you briefly, and you don't have to give a sort of a lecture on quantum biology, or quantum mechanics for that matter, but could you...
Which I actually studied at the university.
Right, I realize that. Could you give a top line on sort of how there may be some connections here between, I think the very small realm of electromagnetism and then the smaller realm of this quantum stuff. I'll leave it at that.
Firstly, those papers are done in collaboration with my colleague from Belgrade University, Dan Rakovic, who is in quantum biology. So he actually incorporated in whole idea of quantum biology, he incorporated resonant recognition model. That's where my role was. But quantum means a small thing.
Yep.
So if you look into resonant recognition model, small thing is a frequency, is one quantum of frequency, because that frequency doesn't happen all the time. The proteins have ability to produce it, but when ATP gives energy to that protein, so it doesn't produce that frequency all the time. It produces only when it gets activated by ATP, which gives it an energy for the electron to go through the protein. And because there are different amino acids on the way, it jumps from one to another quantum state and produce a quantum of frequency.
Okay.
I don't know how clear I was, but that's where quantum biology ties to resonant recognition model. But those papers are actually, my part was only quantum of the paper.
Well, I mean, look, you A, understand it, and B, are making I've always come away, not from your paper, but from other sources on sort of quantum electrodynamics in general with my head spinning. I don't really, it's like, wow, this is an interesting thing in history, but how is this done? How does this happen? And so, hey, if your work links the two worlds, let's say, I think that's an important thing in its own right.
I think I was clear how it all happens.
Yeah, definitely. I want to ask you about sort of mentors, influencers, people that along your path for the last few decades have been influential. In other words, have there been important people that, drove you to continue in this space that, as you said, 30 years ago, people didn't know or believe in, and now, you're very well known. Were there people along this path that said, in other words, if they weren't there, you would have maybe gone into the telecommunications business instead and gotten out of biotech? Are there people along that path that you can you can talk about?
I was in telecommunications and in brain analysis when I met Professor Veljković, who designed this electron-ion interaction potential. And he found that's this energy of free electrons we are talking about.
Right.
And he found there is a correlation with this potential of small molecules with biological activity of small molecules. But he was puzzled how to calculate this potential for long proteins or DNA. And that's actually just in the discussion, he said, for me it looks like a signal. Let's do Fourier transform and find frequencies. And probably if this discussion didn't happen, I will never be in this space that I'm now. He was also my supervisor for my PhD and supported me very much in that work. We published a few papers together initially, and then I went into electromagnetic radiation and stuff. But the first initial one was more like information technology, because you take just those numbers and find the frequency. And I think he was very important in my life. The second thing was when we moved from Serbia to Australia, where I got a lot more opportunities at the universities. And my first job was in biochemistry department at Monash University, even though I was electronic engineer, telecommunication engineer with my basic education. But because I published already quite a few papers, they employed me to do bioinformatics at biochemistry department, but actually they allowed me to do RRM as well. And the first tests and experiments came from this biochemistry department at Monash University. Who else to mention? I think I have to mention the guy from CNRS, Professor Kosmanovic, who was interested in HIV virus.
Right.
So we did in collaboration this work on HIV virus, and he did experiments in France, in CNRS, and the patent is still active and jointly owned by Monash University and CNRS with my name on it, which is really great. But unfortunately, a lot of people don't still believe in it. And it's still hard to get a big support, or not. I'm not talking money, but big support in terms of experiments, in terms of doing stuff. Another person maybe is Bill Pasparialis, who took on this idea very enthusiastically, and we hope something big will come out of this.
Wonderful.
I work now with my husband, Ira Cosic, who has done even the first software for RRM, and along the way he supported me very much in doing this work, in making this software more and more sophisticated as we go away. And now we both run this consulting company, AMALNA Consulting, which is getting more and more successful.
Excellent.
Hopefully more people will take up the idea and we can finally change the paradigm of biochemistry and pharmaceutical industry.
Wonderful. It's nice that you can recognize your husband. My parents worked together for 40 years, and they loved each other very much, but they hated each other at work. So it's nice to see that you can.
Yeah, that's true.
Yeah.
But without support of your actually immediate family,
Yeah,
it's very hard.
Oh, absolutely.
And as I said, my husband supported me from the very beginnings. And he's a software engineer, but now he's becoming more into RRM and biochemistry and learned a lot about this. But he was always behind me to help me with work and also to help me because every research has ups and downs. And sometimes he said, oh, bugger off. No one believes me. I'm going to quit. He said, no, it's a great thing. You can't
It's a great thing. Before we wrap up, I typically the final question on the show, I joke we move from into the science into purely science fiction, but we ask this of every guest. If I had a time machine, a fully functional time machine sitting here with me, and I allow you to go somewhere in it, you can travel to the past, you can travel to the future, you can meet anyone you want, scientists, artists, celebrity, whatever you want to do. Who would that one person in history be? And what do you want to talk with them about?
I think I have my idol in the past, and it's Tesla, Nikola Tesla. Have you heard? You've obviously heard of him.
Yep.
He was also fascinated with resonances. And believe or not, and that's in my book also as a first sentence, I took his own sentence, quoted it. It's that what made organic material from inorganic, it's actually electromagnetic radiation. So he did have some hint of importance of electromagnetic radiation and particularly resonances, because he worked with resonances so much that it is so important for functioning of biological systems, organisms, cells, whatever. So he would be the person I would really love to meet and talk with him. I've written a paper also for a conference, but you can find the full paper on ResearchGate on how his work ties to what I've done, or how he gave me some ideas from his work to do what I've done.
Wonderful. Wonderful. Professor Cosic, it's been an absolute pleasure having you on the show. And, I want to thank you so much for spending time with us. Hearing the stories about people like you that are combining science with sort of the unconventional model of the space, whether that's biopharma or biotech, what have you, is extremely refreshing. And the fact that you are making it happen is additionally refreshing on top of that. For everybody listening, this has been Professor Irena Cosic, Emeritus Professor and Deputy Pro-Vice Chancellor of the College of Science, Engineering and Health at RMIT University in Melbourne, Australia, creator of the Resonant recognition model of macromolecular interactions, founder of consulting firm AMALNA Consulting. It's been an absolute pleasure and honor having you on the show, and just really thank you so much for everything that you do, your ideas, and for joining us today.
Thank you, Ira. That was a pleasure. And just to mention that I'm living science fiction every day.
That's the way to go. I really like that. Thank you so much.
Thank you.